Hello everyone, my name is Alexandra Bernobrandt and I'm a student in the Euro-Mediterranean Master of Neuroscience. Today I'm going to present a project that explores the impact of autoantibodies on the mechanism underlying cognitive impairment in post-COVID patients. Here is the structure of this talk. I will start with the context and the key concepts we need. Then I'll introduce the central questions and the experimental approach. Finally, I'll present the expected results and the ethical framework from the study, before concluding with the potential implication. But before going further into the questions that drive this project, are autoantibodies present in the CSF of PCS patients sufficient on their own to directly alter neuronal function? Post-COVID syndrome affects a significant proportion of patients after infection. Many describe cognitive slowing, difficulties with concentration, and memory lapses. Recent studies have identified in the cerebrospinal fluid CSF of some of these patients autoantibodies. But these patients present also other factors: inflammation, stress, sleep disturbance, vascular impairment, etc. The goal is therefore to isolate the direct effect of autoantibodies on neuronal function. Let's move on the first key concept, autoantibodies. Autoantibodies are molecules produced by the immune systems to recognize and neutralize foreign elements, such as viruses or bacteria. Sometimes these systems become dysregulated. Antibodies start recognizing one's cells as if they were enemies. These are called autoantibodies. They can attack healthy tissues, which can have deleterious effects on the organisms. Another important concept is neuronal excitability. Neurons communicate through electrical and chemical signals. When a neuron is sufficiently stimulated, it generates an action potential, a brief electrical impulse. This impulse then triggers the release of neurotransmitters, the chemical messengers that transmit information to the next neuron. The action potential, AP, is characterized by three essential parameters. The activation threshold, meaning the level of depolarization required to trigger an action potential. The reobars, in other words, minimal current that must be injected to reach this threshold and produce enough depolarization to generate an AP. The firing frequency, meaning the rate at which a neuron generates action potential when stimulated, usually expressed in Hertz. A high firing frequency indicates a more excitable neuron. And the last concept, the patch clamp technique. Patch clamp is an experimental technique that allows direct measurement of the electrical current crossing a neuron or even a single ion channel. Depending on the configuration, one can record the activity of a single channel or of the whole cells. One can also impose a voltage or a current to precisely analyze how the neuron responds. It is therefore an ideal tool to measure the electrical excitability of an isolated neuron. In post-COVID syndrome, autoantibodies present in the CSF could alter neuronal excitability. Two mechanisms are possible: an indirect effect acting on the neuron's environment, microglia, astrocytes, inflammation, metabolism, etc., or a direct effect interacting with the neurons themselves, for example by modifying ion channel function. The central questions become: does the presence of PCS autoantibodies in the CSF on its own directly modify neuronal excitability? We hypothesize that PCS autoantibodies are capable on their own of reducing neuronal excitability by directly acting on the electrical properties of the neurons, specifically ion channel activity, activation threshold, and/or action potential generation. Concretely, in your experiment, the measures of activation threshold, reobars, and firing frequency will be significantly different compared to the control condition. In order to discriminate between the two possible mechanisms, we want to isolate the neuron from its environment and expose it either to CSF from post-COVID patients, experimental condition, or to CSF from healthy individual, control condition. We will use cultured neuron, for instance, human EPCS-derived neurons or mouse hippocampal neurons. And two conditions: neuron exposed to EGF purified from CSF of PCS patients or exposed to EGG from healthy controls. Patch clamp recording in current mode, measure of action threshold, reobars, fire frequency. The advantage of this protocol is that the only factor changing between groups is the presence or absence of autoantibodies. Everything else, temperature, medium, neuronal type, is identical. If the hypothesis is correct, we expect to observe in the PCS autoantibodies condition an increase of the reobars, a higher action potential threshold, a decrease in firing frequency for the same inject current. These changes will indicate a reduction in neuronal excitability, which will demonstrate the direct influence of autoantibodies on the quality of neuronal transmission. The use of cerebrospinal fluid CSF and murine neuronal model raise several important ethical issues. Two regulatory levels must be considered: human ethics and animal ethics. Ethics and regulation concerning human CSF is particularly sensitive, as lumbar puncture is an invasive medical procedure with risks. For this reason, it is strictly prohibited to perform lumbar puncture on healthy volunteers solely for non-therapeutic scientific research. In this project, contaminated CSF containing PCS autoantibodies come from pre-existing clinical samples collected for diagnostic or follow-up purposes. Its use is done through certificate biobanks compliant with European regulation, GDPR, French Public Health Code. Controlled CSF does not require lumbar puncture in healthy individuals. It is residual material from medical procedure or samples already in biobanks. Thus, no healthy participant is exposed to medical risks for the study. No additional invasive procedure for research, confidentiality, and GDPR compliance, proportionate use, strictly justified, and minimal volumes. Ethical transparency. Original patients were informed during clinical procedures and reuse occurs within validated regulatory framework. Approval by Human Research Ethics Committee, CPP, CNIL procedure when data are associated, compliance with the French Public Health Code regarding human biological material. In Europe and France, animal experimentation is governed by directive. All the procedure must comply with the 3R: replacement, reduction, refinement. Replacement is used alternative when possible. In all cases, human EPCS-derived neurons already reduce animal use. Reduction use the minimum number of animals necessary. Refinements minimize pain, stress, and discomfort. Cultures are prepared using standardized, rapidly executed procedures with regulated euthanasia. Murine neurons remain necessary to provide a stable, well-characterized electrophysiological model, allow robust patch clamp measurements, answer the scientific question before potentially moving to more complex human systems. All manipulation, euthanasia, dissection, culture must be performed by personnel certified at levels 1, 2, or 3 in animal experimentation. This project does not aim to reproduce the entire complexity of post-COVID syndrome, but to answer a specific question. Are autoantibodies present in CSF of PCS patients on their own capable of inducing neuronal dysfunction? By isolating neurons from their environment and manipulating only the presence or absence of autoantibodies, this approach allows us to discriminate the specific role of autoantibodies among the many factors involved in patients. If the results confirm a decrease in excitability, this would provide strong mechanistic evidence supporting a link between autoimmunity and the cognitive slowing observed in PCS. Thank you for your attention. I look forward to your questions and comments.